Welcome to the Deep Dive. Today, we're really aiming high. We're not just going over biology basics, we're striving for what you might call elite professional mastery of the core concepts, the absolute foundations of life itself.
Exactly. Our sources lay out this essential framework. Think of it as the 5 key themes that let us understand how living things actually work, how they change, how they interact. It's about getting deep into that structure.
Right. And we want to connect all the dots, from the tiniest atoms right up to the entire biosphere. The goal here is synthesis, building a really sophisticated, joined-up understanding.
So we're looking at 5 main pillars for this framework. There are 4 big unifying themes.
Okay, those are organization, information, energy, and matter and interactions. Got it.
Precisely. And then there's the 5th one, the huge one, the core theme that really pulls everything together, evolution.
It's like seeing biology as this giant layered process, isn't it? Where the really interesting stuff, the big insights, kind of emerge from how all the simpler bits interact.
That's a great way to put it. The complexity arises from the interactions.
Okay, so let's unpack this, starting with theme 1, organization. This isn't just, listing parts. It's about a hierarchy, right?
Yeah.
Where new rules, new properties pop up at each level.
Absolutely fundamental. And nailing down this hierarchy, all 12 steps, is crucial for grasping the sheer scale we're dealing with. It starts really small. At the chemical level, you have the single atom.
Which combine to form.
Molecules, right. And these molecules then assemble into the tiny functional units within cells, the organelles.
And organelles make up the cell itself.
Yes, the cell, which is, as we know, the fundamental unit of life. That's the microscopic foundation.
Okay, so that's the chemical and cellular level. Then we scale up to build the actual organism.
We do. Cells group together to form tissue. Different tissues cooperate to build an organ.
Like the heart or the liver.
Exactly. Then multiple organs work together in an organ system, like the circulatory system or the nervous system. And all of those systems together constitute the individual organism.
But it doesn't stop there, does it? Because organisms don't live in isolation.
Not at all. Interaction is key. So a group of individuals of the same species living in an area, that's a population.
And different populations living together.
That forms a community. Now, take that community and add in the non-living factors, the physical environment. So water, soil, sunlight, air, that whole integrated system is an ecosystem.
And finally, the sum of all ecosystems.
Is the biosphere, all life on Earth and everywhere it exists. So that's the full picture, atom to biosphere, 12 steps. Understanding that scale is step 1 to mastery.
But like you said, the really deep insight here isn't just the list, it's this idea of emergent properties. As you climb that ladder, say from just a bunch of organelles to a fully functioning cell, new capabilities appear, things the individual parts couldn't do alone.
That's where the complexity really explodes. It feels like magic, but it's the direct result of intricate interactions. Think about something like consciousness, or even just a bird's flocking behavior.
Right, you can study one bird or one neuron forever.
But the flock's coordinated movement, or conscious thought, only emerges when millions of them interact in very specific, organized ways. It's not present in the individual components.
And studying this level of complexity, well, it forces us to use different approaches, doesn't it? Sometimes they almost seem contradictory. There's reductionism.
Which is essential, breaking down these incredibly complex systems into their simpler components so we can actually study them, figure out what each part does. You have to do that.
But if you only do that,
you miss the big picture. You miss the emergent properties entirely. So the counterbalance is systems biology.
okay, what's the core idea there?
Systems biology tries to model the dynamic behavior of the whole system. It focuses specifically on how all the components interact, how they influence each other over time. It's like, well, instead of just identifying each instrument in an orchestra, you're trying to understand the symphony they create together.
And weaving through all these levels is a basic principle. Structure dictates function.
Always, at every single level. The shape of a bird's wing enables flight. The structure of a protein determines its chemical interactions. The branching pattern of your airways maximizes gas exchange. Form and function are inseparable.
It all circles back to the cell. You mentioned it's the lowest level that can perform all the activities needed for life. But we need to distinguish the main types, right?
Absolutely. 2 major forms. First, eukaryotic cells. These are the complex ones. They have a true nucleus that holds the DNA, and they have lots of other internal compartments, membrane-enclosed organelles, each with a specialized job.
Like mitochondria for energy, or the endoplasmic reticulum.
Exactly. Think plants, animals, fungi, protists, they're all eukaryotes.
And the other type.
The prokaryotic cells, these are much simpler, evolutionarily older. The key thing is they lack that nucleus and the other membrane-bound organelles. Their DNA is just located in a region called the nucleoid.
And this group includes?
Bacteria and archaea, two distinct domains of life, both prokaryotic.
Okay, so we have the organizational structure down. Now, how does life build and maintain that structure? How are the instructions passed on? That sounds like theme 2, information.
Precisely. Life runs on information. Specifically, DNA, deoxyribonucleic acid. This molecule encodes the genetic blueprint. It carries the heritable information passed from parents to offspring, ensuring continuity.
And within the cell, this information has to be put to use. That's gene expression, right?
That's the core process. Specific segments of DNA, called genes, contain the instructions for building proteins, which do most of the work in the cell. Gene expression is how those instructions are read and executed.
Can you walk us through that mechanism?
Sure. It's elegant, really. First, the sequence of a gene in the DNA is copied or transcribed into a different nucleic acid molecule called mRNA messenger ribonucleic acid. Think of it as a temporary mobile copy of the instructions.
Okay, so DNA to mRNA, transcription.
Then, this mRNA molecule travels to the protein-building machinery of the cell, the ribosomes. There, the mRNA sequence is translated into a specific sequence of amino acids, which folds up to become a functional protein.
DNA to mRNA to protein, the central dogma, essentially.
Basically, yes. And it's remarkably universal across all life. But here's a nuance for that deeper understanding. Gene expression isn't only about making proteins.
Ah, okay. What else?
Many genes are transcribed into RNA molecules that aren't translated into protein. These RNAs perform other vital functions, regulating gene activity, helping build ribosomes, all sorts of things. It adds layers of complexity to information processing.
And dealing with all this genetic information, especially now that we can sequence entire genomes, that's a huge task.
Immense. When we study the complete set of genes for a species, its genome, and especially when we compare genomes between different species, that's the field of genomics.
Which must generate just vast amounts of data.
Absolutely astronomical amounts. And making sense of it all, storing it, sorting it, analyzing sequences, finding patterns, comparing genes requires powerful computers and sophisticated algorithms. That's where bioinformatics comes in. It's the fusion of biology and computer science.
Okay. Structure, check. Information, check. Now, theme 3, energy and matter. To keep all this organized complexity going, life needs fuel and building blocks.
And this is where we encounter a really critical distinction. It's about how energy and matter move through living systems. They behave very differently.
How so?
Energy flows. Chemical cycle. That's the key takeaway.
Unpack that for us. Why flow versus cycle?
Well, think about energy. The ultimate source for most ecosystems is sunlight. Producers, like plants, capture that light energy and convert it into chemical energy stored in molecules, like sugars.
Okay, photosynthesis.
Right. Then, organisms, the producers themselves, and the consumers that eat them, use that chemical energy to power their life processes. Movement, growth, repair, everything. But here's the thing. Every time energy is transferred or transformed, some of it is lost as heat to the surroundings.
Like the heat coming off your body right now.
Exactly. That heat energy can't easily be used to do work by living organisms. So energy enters the ecosystem, typically as light, gets passed along, and eventually dissipates as heat. It flows through, it doesn't come back. It's a one-way street.
Which means life constantly needs fresh energy input, usually from the sun. It's fighting against entropy, in a way.
That's a perfect way to think about it. Chemicals, on the other hand, the actual atoms like carbon, nitrogen, phosphorus, water molecules, they cycle.
Meaning they get reused.
Over and over again. Plants take them up from the soil and air, they get incorporated into organisms, passed along food chains, and then decomposers break down waste and dead organisms, returning those chemicals back to the environment, ready to be used again. It's a continuous loop.
That makes sense. Energy flows. Chemical cycle. A fundamental difference.
Absolutely critical.
Okay, so maintaining this organization, processing information, handling energy and matter, it all requires intricate control systems, which leads us neatly into theme 4, interactions. Specifically, regulation.
Yes, life is full of interactions, from molecules bumping into each other to organisms interacting with their environment. But a key aspect is how biological processes are controlled, often through feedback regulation.
Feedback, like getting comments on your work.
Sort of, but more direct. It's where the output or end product of a process influences the process itself. And there are two main types you absolutely need to know.
Okay, lay them out.
The most common is negative feedback. This is where the accumulation of the end product actually slows down or stops the process that makes it.
So more product means less production.
Exactly. Think of your home thermostat. Room gets warm enough, that's the product reaching a set point. The furnace, the process shuts off. It maintains stability, keeps things within a certain range, like regulating your blood sugar levels.
Right? Prevents things from going too far in one direction. So what's the opposite?
That would be positive feedback. Here, the end product actually accelerates its own production.
So more product leads to even more production. That sounds like it could get out of control.
It does lead to rapid change, an amplification effect. It's less common, but crucial for processes that need to happen quickly and reach completion. Classic examples are blood clotting a cascade, where each step triggers the next much faster, or the contractions during childbirth, which get stronger and stronger.
Got it. Negative feedback stabilizes, positive feedback amplifies. Two sides of the regulation coin.
Precisely. And these interactions aren't just internal. Organisms are constantly interacting with their physical environment, too, taking in nutrients, energy, responding to changes.
Let's pause here for that connection point we talked about.
Hmm.
Think about something you do every day, maybe even right now, texting on your phone. How do these four themes play out just in that simple action?
It's a fantastic example. You've got organization, nerve cells controlling muscle cells, organized into tissues in your fingers and hand, all part of the larger organ systems. You've got information, nerve impulses carrying signal from your brain to your fingers, and visual information from the screen back to your brain.
And energy, obviously.
Yeah.
Your muscle cells are burning fuel to contract and move your thumbs.
Definitely. Cellular respiration, providing ATP. And crucially, interactions. Constant feedback loops. Your eyes see a typo, signals go to your brain corrects your finger movements, you adjust your grip based on sensory feedback. It's all there, a little microcosm of biology.
It really drives home how interconnected these themes are. But none of this happens in a vacuum. It's all shaped by the fifth overarching theme, the big one.
The core theme, evolution. This is the concept that explains both the incredible unity we see across all life forms and the staggering diversity. It's the process of change over time that leads to evolutionary adaptation.
Adaptation meaning?
The remarkable match between an organism and its specific environment. How camouflage helps an insect blend in. How a bird's beak is shaped perfectly for its food source. Evolution, primarily through natural selection, is the engine that produces these adaptations.
And understanding evolution helps us classify life, doesn't it? Based on shared ancestry.
Absolutely. The modern classification system reflects evolutionary history. We group life into three highest level categories, the domains. You have domain bacteria and domain archaea.
Both containing prokaryotes.
Correct. And then domain eukarya, which includes all organisms with eukaryotic cells. Within eukarya, we recognize major groups, often called kingdoms, like plantae, plants, fungi, and animalia, animals, plus various groups of protists.
So incredible diversity. But you also mentioned unity. Where do we see that most clearly?
Everywhere, really, but especially at the molecular level. The universal genetic code based on DNA, the fundamental mechanisms of gene expression, the core metabolic pathways, these are shared across vastly different organisms, pointing to a common ancestor deep in the past.
Which brings us back to that gene comparison. You said prokaryotes might have around 3,000 genes, humans maybe 21,000 or so, but we share about 1,000.
Roughly, yes. And that shared set of genes is incredibly revealing.
Because they must be doing the really essential, basic jobs.
Exactly that. Those shared genes likely code for fundamental cellular machinery, basic metabolic processes, DNA replication, things that any cell needs just to be alive. They represent the deep, conserved core of life, inherited from our common ancestor.
And the process driving both a divergence that creates diversity and the adaptations. That's Darwin's big idea, natural selection.
The primary mechanism of evolution. And to really master this, it helps to break down the process Darwin described into, say, 5 key observations and inferences.
Okay, let's list them out. Number 1.
1. Populations of organisms show hereditary variations. Individuals within a population differ, and many of those differences can be passed down.
2. Organisms tend to produce more offspring than the environment can support, leading to overproduction and competition.
3. Individuals are subject to environmental factors or pressures Predators, climate, food availability, disease.
4. Because of the variations in environmental pressures, there are differences in reproductive success. Some individuals, those whose inherited traits make them better suited to the environment, are more likely to survive and reproduce than others.
And 5, the logical outcome. Over generations, this unequal reproductive success leads to the evolution of adaptations in the population. Traits that enhance survival and reproduction become more common.
We see this play out constantly, right? Like the camouflage example, beach mice whose coat color better matches the sand are less likely to be eaten by predators, so they leave more offspring, and eventually the population's average coat color shifts.
Precisely. Or antibiotic resistance in bacteria. It's natural selection in action. Every species alive today is like a twig on a massive branching tree of life, connected to all other species through this long history of descent with modification from common ancestors.
Okay, fantastic. So we've covered the themes that define life. Now, for true mastery, we need the how do we actually know all this? Scientific inquiry.
Right. The process of science. How we gather evidence and draw conclusions. It relies heavily on different ways of thinking, primarily 2 types of reasoning.
Let's distinguish those. First up,
Inductive reasoning. This is where you go from a set of specific observations to a general conclusion or principle. You notice a pattern in your data, and you generalize from it. This is often how hypotheses are initially formed.
okay, specific observations lead to a general idea. What's the other type?
Deductive reasoning. This flows in the opposite direction, from general premises to specific logical predictions. It's the logic used in hypothesis testing. The structure is typically if-then.
As in, if my hypothesis about camouflage is correct, then I should predict that mice matching the background survive better in this specific experiment.
Exactly. If the prediction holds true when you test it, it provides support for the hypothesis.
This brings us to a really important distinction, one often confused outside science: hypothesis versus theory.
Hugely important. A hypothesis is, relatively speaking, a tentative explanation for a specific set of observations. It has to be testable, and importantly, it has to be falsifiable. There must be some conceivable observation that could prove it wrong.
And this is why science can't really address supernatural explanations, right? Because they generally can't be tested or falsified.
Correct. Science deals with the natural, testable world. Now, a theory in science is something much bigger, much more robust.
And not just a guess or an unproven idea.
Not at all. A scientific theory is a broad, comprehensive explanation for a wide range of phenomena. It's supported by a massive amount of evidence from many different lines of investigation. Theories are so well established they allow us to generate many new, testable hypotheses. Think of the theory of evolution, or cell theory, or gravitational theory.
So much broader scope, much stronger evidence base than a hypothesis.
Absolutely. They're the bedrock explanations in science. Not proven in the mathematical sense, but as close to certainty as we get.
When scientists gather evidence, they collect data. What are the main types?
We often distinguish between 2 broad categories. Qualitative data consists of descriptions, observations using senses, like noting the color change in a reaction, or describing an animal's behavior, the fish swam erratically.
And the other?
Quantitative data. This involves numerical measurements, things like temperature readings, counts, concentrations, lengths. The plant grew 5 centimeters. The reaction rate was 0.2 moles per second. Science often relies heavily on quantitative data for precision and statistical analysis, but qualitative observations are often where new questions start.
And to get reliable data, especially quantitative, the gold standard is often the controlled experiment.
Yes. The key idea here is comparison. You design an experiment with at least 2 groups. An experimental group, which receives the treatment or change you're testing, and a control group, which does not. Crucially, you try to keep all other variables the same between the groups.
So the only difference is the one thing you're investigating.
Ideally, yes. That way, if you see a difference in the outcome between the groups, you can be much more confident that it was caused by the variable you manipulated and not something else.
It's also worth remembering, science isn't done by lone geniuses in labs, usually. It's a community effort.
Very much so. Science is inherently social and collaborative. Researchers build on each other's work, critique it, replicate findings. Integrity and honesty are paramount.
And having scientists from different backgrounds seems really important too.
Incredibly important. Diverse perspectives stemming from different life experiences, cultures, and ways of thinking lead people to ask different questions, design different experiments, and interpret data in novel ways. It makes the entire scientific enterprise stronger and less prone to bias.
Finally, let's define technology in this context.
Technology is essentially the application of scientific knowledge for practical purposes, often to solve problems or create new tools that affect society. Think of developing new medicines based on understanding disease pathways, or creating drought-resistant crops using genetic knowledge.
But the basic science often comes first, right? Sometimes long before the application is obvious.
Absolutely. Much fundamental, curiosity-driven research, just trying to understand how DNA replication works, for instance, might not have an immediate practical application in mind. But those fundamental discoveries are the essential foundation upon which future technologies are built, sometimes decades later.
Okay, that feels like a really thorough circuit. We've hit the organizational hierarchy, the flow of genetic information via gene expression, the crucial energy flow versus chemical cycling, the control through feedback loops, and the unifying power of evolution by natural selection. Plus, how we actually know these things through scientific inquiry.
A complete roadmap for mastery, hopefully connecting those atoms all the way up to the biosphere and the processes that govern it all.
So let's circle back to that gene comparison for a final thought to leave our listeners with. We share those 1,000 core genes with simple prokaryotes, necessary for basic survival. But humans have over 20,000 genes total. What does that vast difference imply?
It's a fascinating question, isn't it? Does it mean that the vast majority of our genome, maybe 95% of it, is dedicated purely to managing complexity? The challenges of being multicellular, having specialized tissues, a complex brain, intricate development. Is it all just complexity management overhead?
Or is there more to it?
Or does that huge reservoir of genetic information hold potential functions we haven't even properly understood or categorized yet? Are there capabilities encoded there, perhaps subtle regulatory roles or responses to rare environmental challenges that are just waiting? What might the next major evolutionary pressure select for, revealing hidden functions within that extra genetic code?
Something profound to think about. What undiscovered potential lies dormant in our own biological instructions?
Keep looking at the world through these 5 themes: organization, information, energy matter interactions, and evolution. You start seeing the patterns, the history, and maybe even glimpse the future possibilities everywhere.
We'll see you on the next deep dive.